Okay, we're back. Now, many of you are going to be working along with like the simple genetics practice worksheet. Okay, so I'm going to kind of focus on that and those types of problems. So if you're just looking at this for, remediation, something else, whatever, it's kind of the same basic stuff. But on the worksheet, the second set of problems, they basically give you 3 possible genotypes. Big B, big B, big B, little b, little b, little b. And above that, it tells you brown is dominant to blue. Talking eye color here, okay? Now, if we have brown and blue allele, we only have 2 alleles, 2 possible phenotypes, brown and blue, and it tells you which one is dominant. So you need to use that information, process it through that leaky brain that you have, right, and then hope it doesn't spill out too much, that you can remember what to do with that. So remember, what is allele? We have 2 copies, 2 alleles. We have brown or blue. One is dominant over the other. Okay, so we can kind of write this down. Big B equals brown. Little b equals blue. All right, now remember, you have 2 copies of every chromosome, so you're going to have 2 copies of each basically gene. So you're going to have 2 alleles functioning here. All right, so the possibilities of people are going to be, you can have 2 big B's, like this guy. Big B, big B. Now it told you, it says, it gives you the genotype, lists the phenotype of this individual. We're dealing with brown or blue. So I'm either going to write in this space, this person has either brown eyes or this person has blue eyes. Since it tells me brown is dominant, then I would write right here, big B is brown. I have 2 copies of big B, which codes for brown. So blue's not even a possibility here. It has to be brown, okay? Now, this person, the second individual, whatever, has 1 big B allele and 1 little b allele. So you have to ask yourself, which one is dominant? It told you, brown is dominant to blue. So even though I have a blue allele, I also have a brown allele, which is dominant over this. So it basically squashes it, beats it down into submission and says, we're going to be brown, get behind me. Okay, that's basically what happens, literally, all right, with the proteins. And so this would also be a brown individual, all right? Little b, okay? Obviously, since I have 2 copies of the little b allele, it's going to be blue. Blue's recessive, so really, this is the only possible genotype of a blue-eyed individual in this case. So here I write blue. And I know that this individual is little b, they have to be blue because of the dominance and recessiveness. All right? Now, on the next part of the paper, it's the exact opposite. Okay, it might say like straight, curly. So it tells you, it lists the phenotype, and you have to write the genotype. It's basically the exact same thing we just did, but let's talk through the dynamic here. All right? In this case, I have S's. It says to use S's. I don't like using S's or C's because they look the same when they're big and little. You know what I mean? So I'm just going to use basically A's here. A equals straight, little a equals curly. Okay? Now, the reason I know this is because my big A usually is going to represent the dominant allele. The little letter is going to represent the recessive allele. It shows me which one is dominant and recessive here. Okay? The way it worked on the last one, this is basically going to be 2 large letters with straight hair. So it's homozygous dominant for straight hair. It has 2 copies of that straight allele. Okay, this is usually my heterozygote, big A, little a. And so I know straight is dominant because having 1 copy of that straight allele gives straight hair. Okay? Curly. Curly is my recessive allele. It's little a. So I know my curly individual is going to be little a, little a. So in this case, the paper's having you write in the genotype of these 3 individuals. They give you the phenotype. All right? Pretty simple. You can use A's, B's, I don't care what letter you use. The only reason I didn't use S's or C's is because they're very hard to distinguish between big and small. Okay? Uppercase, lowercase, capital, whatever. All right? So that should help kind of make sense on the genotype and the phenotype and our possible individuals. Okay? If I'm dealing with 2 alleles, so big A, little a, basically straight, curly, I only have these 3 possible combinations. Homozygous dominant, homozygous recessive, or heterozygous. Alright? So that's basically what that part of the paper is trying to tell you. Also, getting you to realize the difference between genotype and phenotype. Here, think about this. I have 3 possible genotypes. Big A, big A, big A, little a, little a, little a. Those are 3 separate combinations. I only have 2 phenotypes, because both of these genotypes code for the same phenotype because of the dominance. Alright? This little a codes for our second phenotype of curly hair. So there can be differences in the genotype and the phenotype. And that's why we distinguish the 2 with 2 separate words. Alright, next we're going to do some Punnett square work. So we will come back and do the Punnett square work as its own video. Alright, its own podcast. And so we'll kind of talk about the dynamics of Punnett squares and how those work, and percentages. Okay? Later.